Hello everyone. Today I'm going to talk about combined stresses in plants. We had earlier videos in this direction, so I'm sure you must be aware about the combined stress work. Feel free to visit the lab website if you would like to know more about the combined stresses in plants. For today, the combined abiotic and biotic stress tolerance mechanisms in sunflower. This is the topic that I'm going to discuss. Basically, I'm going to present before you one of the manuscript that was published, the work that was compiled out of it. Um, um, the first thing is sunflower is an important oilseed crop, and it's been widely used as a good oil in cookings because of its various compositions, including PUFA. So this is an economically important crop, widely cultivated in India. In this slide, I'm going to explain the relation between the oxidative stress and other biotic and abiotic factors. The sunflower crop is known to get exposed to stresses like cold, drought, salinity, and to some extent, some of the herbicides which are being used to kill the weeds. And some of them are mainly a derivative of methyl villager. So these abiotic stresses contribute to plant growth impact. In addition to that, there are several pathogens, of which the downy mildew causing pathogen is a serious threat to sunflower. Now, all these individual stresses, both biotic and abiotic in nature, have been known to produce free radicals in the tissues, and those free radicals are known to cause oxidative stress. In other words, the point that I am trying to strive here is that these individual stresses can converge in the form of an oxidative stress, at least in part, can contribute to the net damage that individually each one can contribute. In other words, if we can study the oxidative stress related aspects of a plant, can we expand it to see whether the tolerance for individual stresses can also come in? Now, the topic today we are going to discuss is about combined stress. So we also know that the combination of all these stresses that are described here, when exposed in experimental proofs, it's been reported that produces free radicals and also oxidative stress. So this brings us to the point of discussion that the combined stresses also leads to oxidative stress. At least a major portion of the damage that comes to the cell is through the oxidative stress, although the stress factors which are getting combined are all different ones. Now, using this idea, we thought that how about utilizing these convergent points of oxidative stress tolerance as a means to check whether the other stress tolerance can also be derived out of it, which I'm going to talk in a while. So this brings to the aim and the hypothesis behind the study. Can we screen for oxidative stress tolerance, the genotypes, and those genotypes that are identified out of this screening, can they tolerate combined stresses as well? If our hypothesis goes well, then using this, we would be able to come up with a concept that, okay, you screen for oxidative stress tolerance, which is relatively easy to do, than putting many stresses together, which is complex in a large scale setup. So this is going to be feasible. So the aim of the study is to test this proof of concept using the transcriptomic and morphophenomic approaches. If this proof of concept is established, it can pave way for a bigger large scale experiments of using oxidative stress tolerance screening as a method to identify genotypes that are reflective of combined stress tolerance as well. So I'm going to show you the overall methodology that is used in this study and also the hypothesis tested in this study. This slide currently looks complex, but I'm going to show this again at a later stage. By the time you would understand the impact of each of this part. This has basically three parts. Part one is to handle the transcriptomic data sets from public databases. So using different methods, we pulled out the transcriptomic datasets that are available in different platforms were being assembled. Mainly, we found it in 6 different studies in 6 different experiments, and that dealt both biotic and abiotic stresses. The biotic stress is the pathogen that the oomycete pathogen that we are talking about, and this data set was used for meta-analysis. Now, during the meta-analysis, the procedure followed was in such a way that we take two single stress data and find out the overlapping genes, in other words, those that are shared between these two data sets, and again introduce the methylviolin stressed data set to see if how many of them are actually overlapping with the methylviolin stress as well from the shared component. Now you keep doing it for every permutation and combination for all the available data, and you come out with certain number of data sets that can further be utilized for other analysis. This is part one. Part two is screen the genotypes using a compound called menadione, which induces oxidative stress. I will tell you what is menadione in a while. This compound can potentially allow the sunflower genotypes to screen and identify the tolerant ones. Now test the identified tolerant and susceptible ones to see whether they have any relation to the oxidative stress. Now the third component is using these genotypes for transcriptome profiling and merge it to see whether the initially used public data and this transcriptomic data can convey a meaning about its not only its overlap but also whether it is reflective, whether it's successfully identified, whether the oxidative stress screening has successfully identified the genotypes for combined stress tolerance or not. So these three parts have been tested systematically in this study. Since the important component used for compound used for screening in this is menadione, I'm just showing this slide to introduce that. Menadione is a quinone compound. This compound can auto reduce as well as oxidize. Both auto oxidation and auto reduction is possible. What it means is that unlike several other oxidative stress inducers, this doesn't need a trigger. When it is present in cell, it can auto-oxidize as well as reduce, which means that during this process of cycle, this naphtha-quinone can continuously generate free radical, that is the superoxide radical. Now, this is what is shown here. This hydroquinone can get oxidized to become a semiquinone, and the further oxidation leads to menadione, that is a quinone. So this naphtha-quinone can further reduce it back either at single step or at two steps. Whenever there is an oxidation, you can see that there is a production of free radicals, especially the superoxide radical. So now, whenever you introduce this compound into a tissue, you are assured that you have an oxidative stress created without doing any triggers. So you just put the seedlings or tissues into this menadione solution, then you will be able to generate oxidative stress. I'm also introducing a little bit about methylviolin because that was one of the stress that we have considered. This is to simply say that in the mitochondrial electron, the photosynthetic electron transport chain that happens in the chloroplast can be interfered with this compound called methylviolin that can introduce the oxidative stress in the chloroplast, as shown here in this. Now, about 17 years ago, the compound menadione was already shown to impart oxidative stress tolerance, and also the response of the sunflower seedlings has been clearly demonstrated. And more importantly, a method based on how systematically you can use the menadione to screen and to select those seedlings that are recovering to identify tolerant or susceptible genotypes. This procedure simply involves exposing the seedling to the menadione. There are two parallel things apart from control that goes into this experiment. One is a set of seedlings that are pre-induced with the sublethal levels of menadione. Other one that are directly kept into the menadione solution of the challenging concentration, which is lethal to the seedlings. Ultimately, all these three, including the control, have been allowed to recover. The observations on both survival as well as the rate of recovery has been recorded. So this methodology has been already established. So this slide shows the protocol for screening the sunflower seedlings for menadione-induced oxidative stress. As I explained before, the 2-day-old sunflower seedlings are exposed to an acclimation stress, which is a sublethal stress of menadione, and then expose it to the lethal stress of menadione. At that point of time, the seedlings are transferred to a normal condition, which allowed for recovery. And the recovery rate as well as the percent survival has been recorded. And these two parameters were statistically analyzed, apart from some of the physiological, biochemical, and the transcriptomic data that is generated out of it. In a nutshell, this screening has identified two lines, hybrids, that are called KBSH53, which is found to be tolerant, and KBSH42, which is found to be susceptible. The check that is used was a commonly cultivated variety called modern, and this variety is known to be intermediate in response to oxidative stress between these two hybrids. Now, I'm going to tell the actual data that we got from the screening. If you see here, the y-axis shows the number of genotypes, x-axis shows range of survival in the graph that is shown in the left-hand side. You can see that the distribution indicates the presence of genetic variability. You can see there are seedlings of about, there are genotypes of about 12 that survives in the range of 0 to 15, and so on. You can consider for 15 to 30 and so on. And there are genotypes that survives more than 90%, in about 20 number of them, more than 90% survival has been reported. So the main point that I would like to say here in this graph on the left hand side and also on the right hand side top is the left hand side shows the survival and the right hand side shows the recovery growth. The message that I am trying to say here is that there is an existence of genetic variability. Each genotype is behaving differently, which means that there is a possibility that this menadione stress can be used to create, to exploit the genetic variability and to find a line based on its response to this chemical. Now, the middle graph shows that reduction in recovery growth and survival rate. Basically, here the plotting is not only just to consider the survival alone, because survival alone is not enough for a crop plant to be tolerant under the, to be beneficial under the stress, because how much ever the production or productivity that it can make under the stress is more important. If you want to have a good genotype that performs better under drought stress. So the recovery growth also was considered, survival was also considered. You can see that the KBSH42 and 53 are two contrasting genotype, and the modern is somewhere in between as an intermediate, as I told you earlier. So the message from this is that menadione can be used for screening and to get a genotype that can clearly show resistance or susceptibility to oxidative stress. Just to give one example, because we worked with the results, previous results showed only about the abiotic stress. The disease score for the downy mildew was also tested in these three genotype slash the hybrids. It was found that the 42, which was actually susceptible, also was susceptible to the disease. Susceptible in the form of showing higher score of disease, and 53, the KBHS 53 was relatively having less disease score, and modern was somewhere in between, which is again an initial hint to show that the results are falling in line, and that we could clearly get those genotypes that are screened for oxidative stress tolerance are also showing similar trend of tolerance to the downy mildew. Now, just to make sure that whatever the observations in terms of morphophenomic data that we are getting is also reflective at the molecular level, transcript expression profiling was done for a certain number of genes which are known to be reflective of stresses, for example, HSP, and also specifically the oxidative stress, just to make sure that the aspects that are happening in the system are all in synchrony with the phenomorphological data. And it was found that at various plant tissues, this oxidative stress-induced genes and also stress-induced, general stress-induced genes were upregulated. So this again shows that the menadione induces the oxidative stress and broad spectrum disease stress effects. Now, The first thing, as I explained earlier, also concluded we did was the public data analysis using the meta-analysis. The flow diagram is presented here. Basically, the microarray data was collected, data was curated and processed. Using InMex, the meta-analysis was performed after the integrity check, and then the both up and down regulated genes were identified, and then further on validated using the qPCR of all the net identified genes at the after identification using the significance levels of 0.05. Now, the results are like this. The meta-analysis of sunflower transcriptome basically showed that we could identify the commonly regulated genes under abiotic and biotic stresses. And this transcriptomic data can give us a message that we can proceed to use pathogen and drought and also the other stressors as a means to compile the data. And such data is meaningful when it actually screened for the real stresses that are listed in my beginning of the talk. So basically, this graph shows both up and down regulated genes and the unchanged genes. And the middle graph also shows that how each one versus the other stresses performed. Of course, keeping the methyl violagen as a common factor in between when we are comparing the two individual stresses. And we have also compared drought versus pathogen and identified several genes that are up and down regulated. And these genes were later used to shortlist the candidate genes that will be eventually tested in the contrasting hybrids and the genotype that we identified from the screen. Now, I'm reintroducing this overview and the hypothesis just to re-emphasize the point. There are three components to it. We have shown the data for each one of them. The last component, I'm going to show you the data. Now that we have analyzed the public data, that is the first component. Second component, we have screened the sunflower genotypes using the oxidative stress agent, and we also found that the both molecular and morpho phenomic data that we gathered is indicative that the oxidative stress can be used for reflecting the facts around the individual as well as the combined stresses. So that's the second part. The third part, after identifying the contrasting hybrids, those hybrids were tested for both the phenomic as well as the transcriptomic data. So the last segment of the flowchart is going to tell us that whether the proof of concept is establishable or not. Now, for which the genotypes that we have, genotypes and hybrids that we have selected have been exposed to different stresses. Almost all the same stresses that are either involved in the practically relevant concepts or being used in imposing the stress for the transcriptomic data studies. So pathogen, the downy mildew, drought, the salinity, cold, and oxidative stress using the methyl violagen. So these were all either individually, as written here, or collectively in terms of single as well as multiple stresses together. Here, there are more than two combined stresses that were put together at a different stages as sequentially indicated here for the duration that are depicted here. So this becomes the the protocol for combined stress and individual stress comparisons that I will be showing in the subsequent slide. Now, the data that we got from these stressed plants were assessed both in the form of MDA, menadione, and as well as the content of H2O2 that are present in the tissues. The 53, KBHS 53, modern, and KBHS 42 were subjected to the stresses that are indicated in the x-axis. And the left side graph shows the MDA content, and the right side graph shows the H2O2 estimation. To our satisfaction, it was so clear that these genotypes that were behaving the way as like was in the menadione-induced oxidative stress, in other words, the one that are susceptible were also susceptible for the stresses that we are actually exposing here and studying. The one that is resistant was actually resistant to the stresses that we are actually listing it after the experimental result. And modern was in between in many of those parameters. This you can see it both in the form of a menadione estimation as well as in the form of a hydrogen peroxide estimation. So this clearly shows that the oxidative stress tolerant genotypes exhibits tolerance to both the individual, with several of the individual stresses that we have tested, and all of them put together as a combined stress. So this kind of at least phenomic data wise, and it validated the proof of concept, and biochemically also it has been supported. Now, to understand the transcriptomic changes, simply all those genes that as I earlier mentioned that were shortlisted from the public databases have been analyzed in these three hybrids slash genotype for all the individual as well as the combined stresses that have been tested in this study. Part of the data set I am presenting here in the form of a heat map, you can clearly see that wherever the resistant genotype is seen, the induced genes that we expected to have an induction are shown induced, and the suppressed genes were suppressed. So this was very consistent with the data that we expected to be generated from these contrasting hybrids. So this basically concluded us to say that the gene expression analysis under the individual and combined stress can clearly explain the molecular basis for susceptibility and resistance of the genotypes. In other words, the data that we are consistently gathering across the biochemical, phenomic, or the transcriptomic data consistently state that the oxidative stress mediated screening can potentially identify the genotypes that can not only for individual biotic and abiotic stresses but also for combined stresses in sunflower. So this is the inference of this study, and this study I'm sure can provoke a lot of future projects that can aim to use one of these oxidative stress agents depending on the crop plant and depending on the stresses that it is experiencing to screen for, considering that the individual, considering the studies of actual combined stresses being difficult for a large scale screening, this could be an easy option. And more importantly, menadione is an important component that can be used at the seedling level, unlike bigger plants, because the bigger plants, when it is actually started photosynthesizing, only can be used for methyl violation or other oxidative stress inducers because it needs photosynthetic machinery. However, this menadione, since it's because of its auto-oxidation reduction capability, even the non-photosynthetic seedlings, which are very just 1 or 2 days old, can be easily used for screening, which makes it and more high throughput. So this the data, most part of the data were taken from the article that is listed here. And with this, I would like to thank for listening. Thank you.